protein called SIRT1. Now, SIRT1, I'll introduce in a second. It's HDAC activity. Be careful now, it's protein levels, but the HDAC activity of SIRT1 is circadian, and this is now in the liver. This is a different time in vivo of the HDAC activity of SIRT1. The highest activity is nicely antiphasic to gene expression of this specific clock response gene. So it is really nice. Now, what are SIRT1? What are sirtuins? Well, I'm sure that most of you know about this, but the critical thing about sirtuins is that this is a group of 7 HDACs. 2 of them are nuclear, some others are cytoplasmic, and then also mitochondrial. They all have one characteristic that, in my mind, is critical to our work, is that these are a group of enzymes that sense metabolism in the cell, because they use in the cell as a coenzyme NAD. So that function, that enzymatic function of removing acetyl groups is directly sensing or reading the cellular level of energy. I put here a glass of wine, which we all, I'm sure, like, and some of you will enjoy that tonight at dinner. But red wine contains resveratrol, that is an activator of SIRT1, and that activation is part of the French paradox, because SIRT1 has been involved in aging, in controlling fat metabolism. More resveratrol you have, more you'll activate SIRT1, and more you'll be protected from aging and getting fat, basically. All right, so give you an example of what happens when you take now SIRT1 minus cells. That acetylation of H3 at the level of that promoter, that is this clock control promoter, instead to be oscillating now is flat and high. I'm not going to get through all these results, but what we basically found is that SIRT1 works as I will say, a rheostat of the system. It is critical to control the deacetylation of both H3 and BMAL, the other element of the, that is under control of clock acetylation, thereby controlling the deacetylation levels of those substrates. And that results us into a different amplitude in the expression of genes controlled by the clock. So the picture we had only, I will say, 7-8 months ago was the following. We have a picture where the clock and BMAL dimer that are critical elements of the circadian gene expression for all those 15% of the genes controlled by the circadian machinery have a unique function of being acetylating both H3 but also its own partner BMAL. Now we found that clock and BMAL are in a complex. In most cells we looked at and the SIRT1 is responsible for the deacetylation of both the H3 tail but also BMAL. Now what is critical is that we were really tying together the circadian machinery with the metabolism, because SIRT1 reads NAD levels, and thereby the NAD levels, the energy level in the cell, might be controlling the clock. Now, this was the picture until not long ago, and we were kind of satisfied by thinking that 2 enzymes, a HAT and HDAC, will be responsible for the oscillation of gene expression and for the oscillation of acetylation of these 2 substrates. The problem came when we realized that both clock and SIRT1 protein levels, and this is true in most cells, actually do not oscillate. So the enigma was, we have 2 enzymes which do not oscillate, but the readout does oscillate. And this, of course, couldn't work. So the question that Yasu in the lab approached, like, what about the coenzyme? Could be NAD responsible somehow, at least co-responsible for the oscillatory activity of these 2 enzymes controlling so many targets and so many genes? The answer is yes. So you want to look at NAD levels in cells, in culture, but also in the liver, in vivo, and the NAD levels oscillate very nicely. And not only, but the oscillation is antiphasic to nicotinamide oscillation, that is a byproduct of NAD biosynthesis. So this was a first surprise. And of course, this forced me and many people in the lab to go back into really looking carefully at NAD biosynthesis pathways and go back to school, basically. And besides the tryptophan pathways, that is basically what we get from food, probably, you know what, about 10 minutes or something. One critical pathway that controls NAD levels in the cells, the NAD salvage pathway, shown here, is again a notoregulatory loop, another feedback loop shown here. The enzyme SIRT1 is responsible, together with other enzymes like PARPs, to the NAD to nicotinamide transition. Is using NAD as a coenzyme and thereby is exerting its HDAC activity in response to NAD. But then nicotinamide goes back to NAD through the usage of at least another two group of enzymes, nicotinamide phosphoribosyltransferase transferase and these two adenylyl transferases, these three adenylyl transferases. Now, to make a long story short, we know these three enzymes do not oscillate. The question was like, is the NMTP oscillating and thereby responsible for the NAD oscillation in the cell? And the answer is here. Okay, so, well, first of all, if that's true, then we should be able to somehow, if this is true, since SIRT1 is part of the clock, if we block the clock system, we should be able to block the circadian oscillation of NAD, and that is indeed the case with these now cells where the clock is mutated and the oscillation of NAD and nicotinamide is flat. So that told us that the circadian machinery is now responsible for the oscillation in the cell of a metabolite, NAD. And the question was, could be that enzyme, could be the nicotinamide phosphoribosyltransferase enzyme that is now oscillating? And the answer is yes. It's oscillating both in the liver and in mouse embryo fibroblasts. And in both systems, when we knock out the clock system, that oscillation of the enzyme is flat again. Now, a key experiment for this audience is the following, is chromatin IP. We're going to look at the nicotinamide phosphoribosyltransferase enzyme promoter as E-box elements where clock and BMAL can bind. Not only, but the binding of clock and BMAL and SIRT1 to that promoter is circadian. Of all three molecules is circadian. Now, last slide. If all this is true, we should be able somehow to block the nicotinamide phosphoribosyltransferase enzyme, somehow block that if this is oscillating in response for the oscillation, when we should be getting a situation where by blocking the system, we should get a system where SIRT1 is gone, basically. And if that's true, we should be doing this using this particular pharmacological inhibitor. By blocking the enzyme, we should block NAD levels, flatten the NAD levels, and thereby also having a situation SIRT1 is gone. Is exactly what we get. We treat this with this inhibitor, NAD levels are flat, SIRT1 activity is gone, and we go back to a situation where by blocking SIRT1 activity, the oscillation of gene expression and the acetylation of BMAL is now enhanced, showing that indeed what we have is a remarkable situation of two feedback loop. A transcriptional feedback loop, which is the clock system, and then an enzymatic feedback loop, where we have the NAD salvage pathway. Well, we have a situation that is some type of a cat eating his own tail, because we have a SIRT1 responsible for that breeding, that harmonic oscillation of acetylation that I told you about, and for not only BMAL, but also the H3, and for all those 15% of the genes in our epigenome. And now SIRT1 activity is regulated by NAD, and you know that now SIRT1 is somehow involved in this pathway. So SIRT1 is in reality controlling the activity and the levels of its own coenzyme, because NMTP now is oscillating, giving NAD oscillation critical function in the clock, and that feeds back to the clock system right here. So what we're finding is reality system where not only the circadian